My name is Joshua Kwakye-Minott, and I was working with the Kavita Lab this summer in the entomology department. And my main focus of research was the impact of seed corn maggot on the Three Sisters crop system. Go ahead. So a little background. In 2027, New York will be banning neonicotinoid pesticides. And this is very bad for farmers, as many different pests across the state are currently being dealt with by these pesticides. And one of these types of pests are seed corn maggot. They are very bad for farmers, as when they lay their eggs, their larvae hatches, and the larvae goes and they kill the seed before it can even germinate. So this is, of course, very bad for farmers, that they don't know if they'll be getting crop yields this year, that year. So the second part, a little background on the Three Sisters crop system. It is a system consisting of corn, beans, and squash. The corn provides a stalk for the beans to climb up. The beans provide nutrients for the soil, and the squash protects, with the large leaves, protects it from any pests and any weeds. So the study aims to investigate the preference and performance of the Deliapatura or seed corn maggot in these Three Sisters crop system. So our first question was, which of the Three Sisters crops does the seed corn maggot prefer in oviposition, which is the females laying their eggs? So for the first experiment, we set up 3 cups, each with the corn, one with corn, another one with beans, another one with squash. And we put filters on top of the cups for the females to lay their eggs on. And we put them in a cage with 10 males and 10 females so they could breed and then lay their eggs. And the major results from this first experiment is that we found no difference in oviposition choices for the seed types. The low squash value is just, there was a little statistical stuff that happened. After a lot of tests, we found that there's no difference. The second question was, which of the crops does the Deliapatura perform best, like the larva performance? So for this, we wanted to see their pupa weight after they ate the seeds, and they're going to turn into pupa. And we found that there was a difference, a big difference actually. The corn, it had very little performance, had the worst performance, but the other 2, they had similar performance. And then our third and final question was the stand loss, like in a general setting, like say in a real farm. So we put 10 seeds of each in a bed, and we put, again, 10 females and 10 males into the cage, and we just saw the loss that would happen. And actually, very good results is that there was barely any loss. There was, on average, there was around 3 seeds lost in each one, for each one of the crops. So that's very good for farmers to know, so that in the future they don't have to spend so much time developing different techniques to defend against the flies.
Great. Those are super exciting results. Your bioassays look really interesting. Is there anything else? What would you do different about the bioassays if you did it again?
I would definitely try to run some more replicates. A lot of the data, it would have been a lot better if we had more replicates for better statistical analysis. We had a little trouble with some of the results, but yeah.
That is how bioassays and plant-insect interactions go. Thank you very much.